Hello everyone, welcome back. I hope you all are doing good. So today we are going to talk about bacterial cell wall. Okay, let's take lecture outline first. This lecture I'll be delivering you under these headings. Firstly, I am going to discuss about bacterial cell wall and introduction. After that, historical background, overview of bacterial cell wall structure. Then we will be talking in detail about cell wall of gram-positive and gram-negative bacteria. And please note this last heading, peptidoglycan structure and chemistry, this I'll be uploading as a separate video which will be a part 2 of bacterial cell wall. Okay, that you can later on check on the same channel. So now we are going to start from the introduction part. Let's start from the simple heading first. What is cell wall? So always remember, cell wall is the layer which is usually fairly rigid and lies just outside the plasma membrane. So simply if I say what is cell wall is the layer present outside the plasma membrane. So with the help of this picture, you can better know about the location of the cell wall. So this picture is actually showing us a bacterial cell, right? So here what we can observe in the center of the cell, what is there? This grayish blackish filament is representing us nuclear material. Of course, it is not true nucleus. Nuclear material is present without nuclear envelope and it is lying freely in the cytoplasm, which I have shown here by means of yellow coloration. And what you can observe here now, cytoplasm is bound by a membrane, that is reddish pinkish layer, okay, what we call as plasma membrane, right? And now one layer is there which actually surrounds this plasma membrane, or which is actually present outside this plasma membrane, which I have depicted here by means of this green coloration. Okay, this green color layer is actually the cell wall. It is showing us the location of the cell wall present outside the pinkish reddish plasma membrane, right? So our today's talk is actually revolving around this green color layer only. Okay, so let's continue now. What are the key roles of bacterial cell wall? Then always remember, bacterial cell wall in bacterial cell is always responsible for determining the shape of the cell. Okay, what kind of shape a particular bacterial cell is having? It is cocci, means circular. It is rod shaped, means bacilli. It is comma shaped, vibrio, and so on. So these shapes are actually decided by the cell wall, right? So let's see the second important role of bacterial cell wall. So cell wall actually protect the bacterial cell from osmotic lysis, toxic substances and pathogens. Then from this point you should understand that cell wall also act as a barrier to prevent the entry of the toxic substances inside the cell. And secondly, it also add towards enhancement of some virulence properties of pathogens, which help the pathogens to protect themselves from the defense mechanisms of the host, right? So in this way, what we can say that cell wall also contribute towards pathogenicity. Now further we are going to take another heading, importance. So these points what I have covered under this heading will help you to know that how much important cell wall is to any bacterial cell. So first point is this, relatively few prokaryotes lack cell wall. Well, it means cell wall is very important part. It cannot be missed out. That's why majority of the prokaryotic cells, bacterial cells, they are having cell wall. Okay. Second point, let's see now. The prokaryotic cell wall also is the site of action of several antibiotics. So as you know that there are many pathogens, especially bacteria, which are known to cause severe diseases, right? Serious illness or diseases. To overcome those diseases, what we actually use, we go for designing antimicrobial drugs or antibiotics or antibacterial drugs, right? So you should know here that many antibiotics are only having their site of action cell wall, right? What kind of these antibiotics are like penicillins, vancomycin, cephalosporins. These antibiotics are well known to act only on the cell wall of the bacteria. Once they will act on the cell wall of bacteria, what will happen? Cell wall will be damaged or bacteria will undergo killing. So this is highlighting the importance of what? Prokaryotic, or I can say bacterial cell wall. So let's see the next heading now, type of bacterial cell wall. Then you should know here that bacterial cell wall actually we study in two headings, right? Or there are two types of bacterial cell wall. First, cell wall of gram positive bacteria, and second, cell wall of gram negative bacteria, right? After that we are going towards key component of bacterial cell wall, right? So you should know here the key component of bacterial cell wall is peptidoglycan, right? Peptidoglycan, which is also known as murine, right? And it is very unique to bacteria. So now we are coming towards historical background and overview of bacterial cell wall structure. Then you should know that the story of this actually cell wall of gram positive and gram negative bacteria was started with the introduction of a protocol by Dr. Hans Christian Gram, right? In 1884, there was a scientist, Dr. Hans Christian Gram, who developed a gram staining technique or gram staining procedure. Okay, what actually he observed when he used the same procedure, gram staining procedure, to stain different type of bacteria, he observed two different type of responses, right? Protocol was same, but observations were different for different type of bacteria. What actually happened, let's see here with the help of this picture. One group of bacteria were found to take the color of a dye which we call as primary stain crystal violet, and they appeared purple or violet color. And if we talk about another group of bacteria, using the same staining procedure, they were not stained by crystal violet, but they were stained by a secondary stain which we call as counter stain safranin and appeared pinkish in color. At that time, the bacteria what took the crystal violet color and appeared purple, they were called as gram positive, and another group of bacteria which were going to take the color of safranin and appeared pink, they were called as gram negative. But do you know at that time uh, what was the nobody was clear about it actually. What was the difference, structural differences between this? Let's continue this slide now, then you will come to know what are the true structural differences between above two groups. This was the question at that time. If we are going to apply same staining procedure, then why results are different? Why all bacteria are not going to take the same staining reagent color? Why it is so some are purple and some are pink? Then at that time it was actually not clear what are the two structural differences in two groups. Why this kind of variation is there in the results and observation, right? So next you should know how this problem was actually resolved. So when and how it became clear, then you should know with the advent of a technique known as transmission electron microscopy. You should know that electron microscopy technique is actually including two types of microscopy, SEM and TEM. SEM stands for scanning electron microscope that helps in just surface examination of microbial cells, and TEM stands for transmission electron microscopy that help to study the ultrastructural analysis or ultrastructure of any bacterial cell. So through the advent of after the advent of this transmission electron microscopy, it was known to everyone at that time that actually there lies some differences in the structure of their cell wall. That is the reason two group of bacteria are giving us different type of results when we are going to use the same staining procedure to stain them. So I hope now you are very much clear about it, that from where the story of this gram positive and gram negative cell wall, they are different from each other, they are having some different nature or different characteristics actually got started, right? So now we are moving ahead. Gram positive and gram negative bacteria cell wall overview, right? So here we are going to talk about the first the overview of the structure. Okay, then one by one, we will be comparing, of course, as well as covering the salient features, which will be important for your notes making and, of course, for examination attemptation, right? So let's make your understanding first clear from here. Here I have drawn a line, and on one side I'll be telling you about I'll be giving you an overview of gram positive bacteria, and other side we are going to take an overview of gram negative bacteria, gram negative and gram positive bacteria what cell wall, right? So let's see now. This picture is showing us gram positive bacteria cell wall. So what you can see here, simply this pinkish layer is representing us what, plasma membrane, and outside this plasma membrane this orange layer is showing us the presence of what, cell wall. Cell wall, we all know it is made up of what, peptidoglycan, right? Now we are coming towards gram negative bacteria. Let's see the overview of this gram negative bacterial cell wall. So here again we can see this pinkish layer is representing us what, plasma membrane, and outside this plasma membrane we know this thin layer here is showing us what, peptidoglycan. Okay, you can also see the labeling, and the outermost layer which is present outside this peptidoglycan layer, one additional layer is present here, that is outer membrane, which was not present in case of gram positive bacteria, right? So this is the overview of just the structure of gram positive and gram negative bacterial cell wall. So one more thing you can see here, the periplasmic space, right? Between the two membranes, the space what is present, that we call as periplasmic space, that I'll be further telling you. Now we are just going to see the same points what I have tried to show you here with the help of this text. Now you can see peptidoglycan in case of gram positive bacteria, it is the single layer, and of course it is thick. Okay, when we compare it with gram negative bacteria, and thickness is around 20 to 80 nanometer, right? For peptidoglycan layer or murine, why we call it is as murine? Because it is a polymer of sugar and amino acids, right? And of course, lying outside the plasma membrane, we are clear about it. Now you see for gram negative bacteria, what text I have included here, it is quite complex. Quite complex when we compare it with the gram positive, right? So here what we can see, peptidoglycan layer is 2 to 7 nanometer thick. When we compare it with gram positive, then it is very much thin, right? It is only 2 to 7 nanometer. And on the other hand, in case of gram positive, it is 20 to 80 nanometer. Please note, if you are having MCQs or you are preparing for any microbiology entrance examination, then this is very important question. What is the thickness of peptidoglycan, right? So in case of gram positive and gram negative bacteria. And second, why it is complex? Because here this thin peptidoglycan layer is also covered by a outer membrane, right? By an outer membrane which is 7 to 8 nanometer thick. So this outer membrane is actually absent in case of gram positive bacteria. So I hope you got the overview now, है ना? Still some differences are there in chemistry of both cell walls, but these are the major differences what we can understand with the help of this overview slides, right? So you see some important points I have also included here, right? Which generally students found, and at that time terms should be clear to them. So I have included as a note. If you want to skip, you can also skip. So firstly, cell envelope. Cell envelope is the term what we use for all the structures present outward from the plasma membrane, right? It includes plasma membrane, cell wall and capsule. Capsule, which is not present in all bacteria, it is a mucilaginous substance, slimy layers, which are made up of proteins and carbohydrates, right? But they are not present in all bacteria. So all these actually form a part of cell envelope. Second point you should know here, periplasmic space. What I have shown you here, periplasmic space is actually the space which is present between the plasma membrane and the outer membrane. Okay, in case of gram negative bacteria. In case of gram negative bacteria, please note what is the periplasmic space? The space present between the plasma membrane and the outer membrane. That is called as the periplasmic space, and always remember it is very much dominant in case of gram negative bacteria. On the other hand, if we talk about gram positive bacteria, in case of gram positive bacteria, the space present between the plasma membrane and the peptidoglycan layer, that is called as the periplasmic space. But periplasmic space is every time not that much prominent and significant in case of gram positive bacteria. Rarely it can be observed, right? Next, we should know periplasm. What is periplasm? The substance that occupies this periplasmic space, that is called as periplasm. And you should know in periplasm there are many type of proteins which we call as periplasmic protein, which are helpful to bacterial cell in many ways, what I have further included in upcoming slides, right? So now we are coming towards structure of gram positive cell wall and structure of gram negative cell wall. So here we will first have a look on the gram positive cell wall structure and gram negative cell wall structure with the help of diagrams, right? So now you will see here the kind of labeling I have made here to show you. One by one we will understand what kind of layers are there, okay, which will help us to understand the structure part. Let's start. So this layer is representing what here? This is actually representing the plasma membrane, right? So let's see what kind of plasma membrane components are shown here in this picture. This is peripheral protein which is present on the periphery, and these type of proteins, these are called as the integral protein, which are present on the inner side of the plasma membrane. Then always remember that plasma membrane is actually in case of bacteria is fitting same kind of model, that is what we are studying from our school time, given by Singer and Nicholson in 1970s, fluid mosaic model. So according to fluid mosaic model, same kind of properties are having bacterial cell wall, bacterial cell membrane is having, right? Means peripheral protein, integral protein, phospholipids, carbohydrates, right? So these are the constituents which make the plasma membrane. So after that we are coming towards the topmost layer. Of course, here you can see many layers are there. These layers are representing what? These are representing the key component of bacterial cell wall, that is peptidoglycan. In case of gram-positive bacteria, so of course it is also representing somewhat thickness of the peptidoglycan here. That it is the thick layer of peptidoglycan is present over here, right? So many peptidoglycan subunits are cross-linked with each other to form thick layer of peptidoglycan here, right? And here now you can see this space which is present between plasma membrane and the peptidoglycan, that we call as periplasmic space, right? And now we are coming towards another chemical substance which is present in the gram-positive bacterial cell wall, and that is teichoic acid, right? Teichoic acids are also responsible for providing bacteria negative charge, right? Negative surface charge, we all know bacterial cell surfaces are carrying. So teichoic acids are also responsible for contributing the negative charge on gram-positive bacterial cell, right? And now you can see teichoic acids, they are actually present where? They are attached to the, they are linked to the peptidoglycan layer, and some teichoic acid molecules are also embedded where? In the plasma membrane. When they are attached to the membrane lipids, then they are called as the lipoteichoic acid, right? Now we are coming towards gram-negative cell wall. So again we are going to start from the plasma membrane part, right? Same kind of proteins, peripheral protein, integral proteins are there. Let's see the next part now. Peptidoglycan, right? Here peptidoglycan is present, but again if you will be comparing with gram-positive bacteria, what you can observe? Peptidoglycan layer here is thin, right? It is not that much thick, which we have also earlier learned in the overview part. And now we are going to observe the next part. What is actually this? This is indicating again us the presence of membrane, because we can see here phospholipids are there, right? So this is the outer membrane, and outer membrane in this case is chiefly made up of a component what we call as lipopolysaccharide, LPS, right? Let's see the next labeling, and here you can see between the outer membrane and the plasma membrane there is a space, okay, which is also covering in between the layer of peptidoglycan. This space we already are knowing about it, we have discussed. This is periplasmic space, right? And now we are coming towards some other parts, some other components of the gram-negative cell wall structure. What these filaments are indicating us? These are indicating us, these chains are indicating us the presence of O side chains. O side chain is a part of actually what? It is a part of LPS, lipopolysaccharide, right? Next thing you should know, Brown's lipoprotein, right? These are Brown's lipoprotein. These are a special type of protein which are absent in case of what? Gram-positive bacteria. Similarly, LPS, outer membrane, these are also absent in case of gram-positive bacteria. So parallelly, you can also look towards some of the differences between the structure of gram-positive and gram-negative bacteria here. Now coming back towards Brown's lipoprotein. So what you can see here, this brown lipoprotein actually act as a adhesive site, right? Contribute towards adhesion of peptidoglycan layer with the outer membrane, right? So in this way, this is also present over here. Next, you should know porine. Porine includes a very special class of protein which is also present in case of outer membrane of gram-negative bacteria. Porins are also known to support the transport of some special type of molecules across the membrane. And secondly, you should know here porins also add towards the microbial drug resistance, okay? Well, this is another point. So these are the some of the structural differences what we have, structural differences, or we can say structural points, what we have understood with the help of diagrams. Now we are coming towards salient features of each of these type of cell walls, gram positive and gram negative, which will also be helping you in getting some notes for attempting your exam, right? So now I have included salient feature of gram positive bacteria cell wall. So let's see here, already the same picture I have included here, pasted here. So here you can see peptidoglycan, right? Peptidoglycan, what is the thickness of this? 20 to 80 nanometer, right? Peptidoglycan, I will upload it separate video on peptidoglycan chemistry, but again, just to introduce you, here I have also mentioned a little bit chemistry of peptidoglycan, that it is made up of two subunits, NAG and NAM. NAG stands for N-acetyl glucosamine and NAM stands for N-acetyl muramic acid, right? So these are actually the sugar derivatives, right? Which form the main subunit of the peptidoglycan. Okay, and also tetrapeptide chain is present in the peptidoglycan. Again, this is a part of structural chemistry of the peptidoglycan, which will be a part of our another class. Right now, you just need to know here how many sugar subunits peptidoglycan is having, NAG and NAM. What is the peptide chain length? Of course, it is having 4 amino acid tetrapeptide chain. What is the thickness? 20 to 80. And of course, it often contains peptide interbridge. Again, this peptide interbridge understanding you can better understand when we will be covering peptidoglycan chemistry, okay, which is present in case of gram-positive bacteria, right? But this peptide interbridge is absent in case of gram-negative bacteria peptidoglycan cell wall. Let's see the next point now, teichoic acid. Teichoic acid, I have already told you, it is usually present in large amount. Covalently, it is connected to peptidoglycan and plasma membrane, as I have already told you, to peptidoglycan, teichoic acid, to plasma membrane, we are saying it lipoteichoic acid, right? It extends to the peptidoglycan surface, right? Being negatively charged, give the cell wall its negative charge. Polymers of glycerol or ribitol joined by phosphate groups. This is the chemistry of teichoic acid. Okay, teichoic acids are made up of what? So you see each and every point what I have mentioned here in teichoic acid, peptidoglycan, they are actually the answers of single MCQs which are used very commonly asked in many entrance examinations of microbiology, right? So, and competitive examinations. Let's see the next point, periplasm. Periplasm, I have already told you, right? It is the space actually between plasma membrane and the peptidoglycan layer. Then always remember, lies between this contain few proteins usually attached to plasma membrane. So what is there in periplasmic space? The substance what is present, what that we call as periplasm, and in periplasm some proteins are there. Those proteins we call as what? Periplasmic proteins, right? Those periplasmic proteins generally remain attached to the plasma membrane. So this is the periplasm chemistry. Let's see the last heading, surface proteins. So you should know here that cell wall of gram-positive bacteria also produce some surface proteins which are involved in actually the interaction of the cell with its environment, right? Binding to peptidoglycan, teichoic acid or other receptors either covalently or non-covalently. So what I mean to say here is that cell wall of gram-positive bacteria are also known to produce some surface proteins, and those surface proteins help the bacteria to interact with the with its environment. But that environment can be host, that environment can be soil, environment can be water where the bacteria is present, right? And of course, where those surface proteins are attached, if they are produced by bacterial cells, they are actually bound to peptidoglycan, teichoic acids or other receptors by covalently or non-covalently. Some of the examples of such surface proteins include S layer and M proteins of pathogenic streptococci, right? So these proteins are contributing generally towards pathogenicity or called as somewhat virulence factors for bacteria, right? Which enhance the pathogenicity of bacteria. Like M protein are very well known virulence factor for streptococci in order to cause the disease caused by streptococcus. So now these were the salient features of gram-positive cell wall. Now we are coming towards salient features of gram-negative cell walls. Again, you can see same picture I have taken, what I have shown you earlier, right? For gram-negative cell wall. Now we are going to take one by one the points for this. So peptidoglycan layer, already it will be having NAG and NAM along with tetrapeptide chain as for gram-positive, right? Key component is same for gram-negative cell wall. Then if we talk about thickness, then you should know 2 to 7 nanometer thickness is there, or we can say when we compare it with gram positive, then of course peptidoglycan layer is thin in case of gram negative bacteria, right? Next point, you see here outer membrane. Outer membrane lies outside the peptidoglycan layer, right? This is the peptidoglycan layer, this one, and outside this outer membrane is present, right? So linked to the cell by Brown's lipoprotein and many adhesion sites. Already I have told you this Brown's lipoprotein help the outer membrane to get attached to the peptidoglycan layer, right? And now you should know outer membrane is made up of lipopolysaccharide, what we generally call as LPS. Okay, so expanded form I have not written here, I forgot to include. LPS stand for lipopolysaccharide, right? So LPS is further made up of three subunits: lipid A, core polysaccharide and O side chain, right? So lipopolysaccharide of outer membrane further plays very important role for bacterial cell wall. It helps in production of endotoxin, It helps to provide bacterial cell adhesion. It helps to provide bacteria to enhance their virulence and also act as permeability barrier. So what you can see here, that outer membrane, it's mainly responsible for enhancing or improving the pathogenicity of bacteria. That's why you should know that most of the gram-negative bacteria are pathogenic when we compare them with the gram-positive bacteria, because of the presence of LPS in the outer membrane, right? And you know, in LPS, lipid A, core polysaccharide O side chain, what act as endotoxin? Then you should know lipid A is actually the that portion, or we can say that subunit of the LPS, which actually codes for endotoxin, right? And core polysaccharide, it is also made up of sugars, and around 10 unusual type of sugars are present in core polysaccharides, okay? And O side chain, it is again of proteinaceous nature, and when somebody develop vaccines or some kind of drugs to for acting against gram negative bacteria, then if they are antibody based, then O side chain they are treat as antigen, right? So you can forget this point if it is not required. O side chain actually act as an antigen against which, if somebody is going to develop drug, they develop antibodies, right? Then bacteria will be neutralized. So now we are going towards porine proteins, right? Porine proteins are also present in gram-negative cell wall, right? Porine proteins also help the bacteria again in some other way. How? It helps the bacteria in permitting the passage of some small molecules like glucose and other monosaccharides. Okay, the transport of or the passage of such kind of small molecules, otherwise it's not possible through the plasma membrane. So in this way, porine is further an added advantage to the gram-negative bacteria through its cell wall. So next is periplasm, right? Periplasm. Already you are now knowing about periplasmic space. Periplasmic space is the space lying between outer membrane and the plasma membrane, right? And you know, there is a substance which occupies this periplasmic space that we call as actually periplasm, right? And periplasm is chiefly made up of, or we can say it is well known to contain various kind of proteins, what we call as periplasmic proteins, that help the bacteria to acquire various kind of nutrients by means of production of hydrolytic enzymes and transport proteins. So these are some of the salient features of gram-negative bacteria cell wall, right? And now, of course, it is very important part. Without this topic, bacterial cell wall topic is never completed. This is peptidoglycan structure and chemistry. So these 4 slides will be a part of our second video. So stay tuned. I hope this information will be useful for all of you. Thank you so much. Stay connected.